All right. Thank you all for staying to the last talk. Someone has to go last, so I appreciate that. So this is a work that we've been trying to get published for a while, actually. So it's a preprint, and this is a very short talk. So I'm really just going to skim over the highlights. So one of the goals that we've been working on for a while is using health records data to discover drug combinations impacting cancer incidence. So why are we looking at drug combinations? So actually, if you look at health records data, and if you just know anything about places like the United States, most people who are taking one drug are actually taking more than one drug. So people are commonly exposed to drug combinations. And there's some really interesting examples of drug combinations that have been associated to changing cancer incidence from either epidemiological or experimental studies. So this little diagram summarizes the hallmarks of cancer. So those are different abilities a cancer must gain to grow. One of these is death resistance. And estrogen by itself is known to possibly reduce the risk of breast cancer. But if you combine estrogen with progesterone, those two drugs together increase the risk of breast cancer by triggering a different hallmark of cancer, the pro-growth pathway. Another drug combination, an antidepressant and an anticoagulant, those two have been shown synergistically to possibly induce autophagy and overcome death resistance in cancer cells. So that was experimental work. So I think these are really exciting examples because the estrogen and progesterone example, that finding probably resulted in preventing a lot of cases of breast cancer, stopping people who are at risk of breast cancer from taking that. And the other combination I mentioned could be a drug repurposing potential for glioma in that example. So how could we discover drug combinations impacting cancer incidence? So what we could do, what we should do in a perfect world is run a randomized clinical trial to discover this. And this is how it might work. You might enroll people into your clinical trial based on, so this is for an arbitrary combination drug one and drug two. So we might enroll people who are taking a drug one and then randomly assign some of those people to take the drug combination with drug two and the other people to keep taking drug one alone. So, and then you follow up the two groups that were randomly assigned, so there shouldn't be any biases. Follow up and see which of these get cancer more than the other. So that's very conceptually simple, but the problem is that's extremely expensive. And we can't just do this randomly with combinations of drugs because we have to do this for every possible combination of drugs, of which there are thousands. So what we're trying to do is use observational data to simulate this clinical trial for any possible pair of a drug one and a drug two. And once we have that method, we could just rerun it for all the possible combinations that are represented in our data and see what we get. So here's what the data looks like. This is one person again. And the observational data, we're using claims data, is just a timeline where this person takes drug one and then they have a diagnosis and then they take drug two and then they have cancer. And you have this data for millions and millions of people. So that's what it looks like. And basically what we can do is say, which of these people are taking drug one? All right. And those are the subset taking drug one. And then you look of those people, which of them match our other eligibility, like they don't have a history of cancer or taking drug two. So then we keep only the people who would be eligible for a clinical trial. And then we just say, all right, so some of these people took the drug combination and others did not. We have two cohorts. We compare their cancer. So that's what you could do if you wanted to get a lot of biased results. So this is what I'd call the naive estimate. And the problem is that this is not randomized data. This is not a clinical trial. This is observational data. In the real world, people took the drugs they took because of their health history. So let's say this drug two here happens to be a drug for menopause symptoms given to people with menopause. And this diagnosis is a diagnosis of menopause. So that is a classic example of confounding. So people after menopause are more likely to get breast cancer. And so anyone who's taking a menopause drug is probably at higher risk of breast cancer than other people. So we would find some association without there actually being any effect of the drug combination. So here's where I'm just really going to skim over a lot. So what we try to do is remove this confounding. So what our assumption is that anything that happened in your past can influence both whether you took a drug or a drug combination and whether you have an increased risk of cancer. So we say all of your medical history influenced the drugs you take at time one, time zero, your diagnoses at time 1, and all of this influences whether or not you got the drug combination. And furthermore, all of that influences whether or not you got cancer. So out of all of these numerous mess of arrows, the only arrow we care about here is whether the drug combination influences your risk of cancer. So we want to remove all of these other arrows. And how we do that is by modeling the probability of someone taking the drug combination given their health history. And there's a number of ways of getting that probability of taking the drug combination. And then once you have that, you have very well established methods called marginal structural model weighting. So here you can see that in the drug combination cohort, this confounder is more frequent than it is in the drug one only cohort. And but after weighting, you've removed that association, and then you have an equal level of the confounder in the two populations, etc. Then you can do your weighted Cox regression on the weighted population and get your association. So as a result, we're able to execute 9,000 drug combination association studies for 9,000 different drug combinations. And we do it for all of the common types of cancer that we're able to do it for. So here each point is one drug combination effect. So the x-axis is before we try to adjust for confounding. So a higher log ratio on the x-axis is more harmful, like that predisposes you to cancer, and the more negative would be it putatively prevents cancer. And so there's a number of interesting things that come out of this. If you compare the axes, you can see the axes are really different. There's this issue, this artifact I don't really have time to talk about, but on the x-axis, you can see that, if you compare the x-axis and the y-axis, the x-axis shows a lot of confounding that's reduced by adjusting for confounders. So because this point on the x-axis is much higher before adjusting for confounding versus after, but there's still a big issue of unmeasured confounding. And I think this linear relationship between before and after adjusting for confounding is able to kind of reveal the level of unmeasured confounding, which is a different research direction we're taking in the lab. But for now, the proposed solution is we're making a hierarchical model that looks for what types of drug combination effects are consistent across cancers of multiple sites. So there's examples of cancer effects, because we know cancers share all these hallmark abilities, and things like aspirin have been shown to reduce risk of cancer in multiple types of sites of cancer. So we want to see, do we find any drug combinations that are associated with changed risk of breast cancer, lung cancer, et cetera? And we also look for effects that are shared across drugs sharing an active ingredient. And I don't have time to talk about this, but we also contrast to other negative controls. So as compared to previous drug cancer findings, there's not really a gold standard, is another difficulty of this project, but there are some previous drug-wide association studies of cancer. So this study systematically did, again, a drug-wide analysis. They found 21 associations they focused on, and another drug-wide study included 15 of those and was able to replicate 10 of those 15. That's 14, not 15. And we had 15, and we were able to replicate 10 of the 15. So I think that this is fairly encouraging, because despite the real, we're looking at drug combinations, we're trying to adjust for all this confounding. It's very different. But despite the big differences in study design and goals and everything like that, we have a comparable replication rate to previous work. So the preprint is available, and here's all my contact informations, and I'll take any questions. We don't really have much time. Sorry.
Thank you very much for the very nice talk, Rachel. So we have a question.
Thank you. Thank you so much, Rachel. I was wondering, first, very quick question. How come you are modeling instead of much more simple propensity score approach?
Mm-hmm.
And then also the second question, to validate the interacting drugs that you have found, like you were mentioning,
mm-hmm.
like one approach that you use. Have you potentially thought about checking for whether the drug is also as targeting a protein that maybe may have protein-protein interaction? With,
if the two drugs.
Exactly. Maybe using knowledge-driven databases out there of drug target or protein-protein interaction to also validate what you're finding.
So for the first question, we are basically doing propensity score methods, I would say. The only difference, and I really didn't have time to talk about it, is is we're dealing with the time issue. So that's something that makes it different from like a straight propensity score. But marginal structural model is basically using a propensity score, but is a bit more complicated, I would say. For the second one, I actually, in order to satisfy reviewers, I searched far and wide for some sort of validation that I could do that wouldn't be like 2 papers in one. And I really think that's a separate paper, unfortunately. And there are some methods that use like network analysis to try to predict drug combinations that seem fairly successful. That could be a decent validation, but yeah, I just felt like it was too much to put in one paper. Thank you.
Thank you. I was wondering if you looked at drug combination screens in cell lines and whether you found any relation to your real life data or not with those, since it's always controversial how realistic are cell lines.
Right. Well, I also did not find really great data on that too. Again, to satisfy the reviewers, I really was looking, and most drug combinations I found in cell lines are looking at cancer drugs. So they're trying to look at like a combination of like 2 known cancer drugs to see if if it kills the cell. And there just wasn't good data on like normal drugs like antidepressant or an epileptic drug or something like that. So yes, I think that would be great, but there just isn't good data as far as I know.
Great talk. So were you thinking of adding genetics into this, of people who are predisposed to like getting something like breast cancer or any of that?
Yeah,
And
like
would
as a
you-
confounder?
Yeah, as a confounder,
Yeah.
and then looking at drug-to-drug interaction, because if they have a mutation, then it could be like not just the drug, but also but adds more complexity.
Yeah. So I don't know if genetics would really be a confounder unless there was a genetic. So it would have to be genetics that influences both your risk of taking the drug and your risk of cancer. But there could be some examples of that. That would be easy enough. But for drug combination studies, you really need huge data because you need to have data on people who took drug 1, then drug 2, then got cancer. This data has like 100 million people, and there's no genetic data that's as big as that. But there will be like years from now, and I think that would be really cool.
Thank you.
We have one online question
Oh.
as well. Does tamoxifen protect from cancer incidence for different cancers?
Oh, for other cancers? Well, you don't know because the person's not here. So I'm going to assume, sorry to whoever that is, they're asking like, if you take tamoxifen for like breast cancer prevention, would it change your risk of developing other cancers? And I honestly probably would not include tamoxifen in the study because if somebody is taking tamoxifen, maybe they already have cancer. So there probably wouldn't be that many people included. I don't know the answer to that, but I think it would be interesting as maybe a follow-up to do. You could do the same thing with combinations of drugs and not cancer incidence, but cancer outcomes. And you could look and see if people who are taking tamoxifen for their breast cancer treatment might have a changed risk of other cancers down the line. So that could be something interesting. Feel free to get in touch, whoever that was. Thank you for the question.
Great talk, great methodology. Could you give us a little insight into what you actually found in the end? What were those drug combinations that maybe prevent cancer or the other way around?
So I do have one slide that I put on here. There's one. Okay, here's one that I thought was really cool. So, well, on the left is an example of something that was confounding that we were able to basically exclude with our hierarchical model. Which I won't go into because I know I'm out of time. On the right, this one was very interesting to me. So a combination of omega-3 fatty acids and fenofibrate was found to reduce. So the x-axis is the log hazard ratio, and the orange bar that goes across is our confidence interval after pooling across all of the different cancer sites. So that is like our like top interesting candidate. Some other examples were also statins we found, which is kind of a known thing that statins are known to possibly reduce risk of cancer. But this one was really interesting because both of these might have a synergistic effect on lipid processing, which could possibly impact cancer via cancer has very high metabolic requirements. So that's one example.
So it also looks like there is an effect size here that's measurable. Is it more than for single drugs alone or in the combination? Are you finding synergy somehow?
Yeah, so the
or-
synergy, I mean, so that is kind of measured against the single drug alone. So the effect size is actually comparing omega plus fenofibrate against people who just took omega-3 fatty acids alone. So that's the effect size we're measuring.
Okay, so there is inherent synergy in here. 'Cause
Yeah.
You're comparing to. Okay,
Yeah,
see.
that's the idea. Yeah.
Interesting.
Yeah.
Great work.
Thank you.